less stronger association. And that just means, when we say an association, that means in the literature thus far. It doesn't mean that they're not major players. So again, we've really expanded our concept that mast cells can play a role, not just in allergy, but in all kinds of other conditions. And that has led to this notion of mast cell mediator disorders. Some people call this mast cell activation syndrome, MCAS, but the immunology literature, and this is from an article by Dr. Theo Herides, who really is the expert on mast cells, says mediator disorders is a better way to put it. And what are these conditions? Well, they're situations where there's an inappropriately elevated serum or urine level of any mast cell mediator. So it could be histamine. In some cases, it's not histamine. It could be tryptase. So, or it could be one of any other mediator. So the mast cells can be selective, as I said. And these mediators are elevated in the absence of a comorbidity that could explain such an increase, right? So the mast cells are overreacting. Now, they're still reacting to the same kind of stimuli, right? But they react in a way that's wildly inappropriate. In other words, without exposure to a bee sting or penicillin or pollen or typical allergic triggers. They might just be reacting to a normal food. And when they react the way you would expect them to react, with flushing, itching, low blood pressure, diarrhea, headaches, irritability, malaise, memory problems, neuropsychiatric issues. So as you can see, this can have an overlap with a wide range of conditions. And it's definitely a scenario that you want to consider when somebody has a chronic non-explained illness. It's definitely a workup that I will engage in with a number of patients. One thing that I want to mention here is that one of the best ways to stabilize mast cells is with a compound called quercetin. Quercetin is a yellow pigment, a flavonoid, that's found in fruits and vegetables. It's highest in red onions and apple skins, red apple skins in particular. And it's been known for many, many years that it has anti-allergic and neuroprotective activities. It's also known to inhibit the release of histamines from basophils and mast cells. Okay, so the question is, well, how does it do that? It's thought to stabilize mast cell membranes, but it probably has an impact on the DNA of mast cells as well. And those mechanisms are just beginning to be elucidated. So we're going to come back and talk about quercetin later in the course more than once, because it does appear to be a very, very useful compound for people with mast cell disorders, as is a related compound called luteolin. So this is just one example of how understanding the molecular mechanism of what's going on can lead to an intervention that is based on foods, or perhaps an extract of foods in the case of quercetin supplements. This is how the system works. Now I want to talk about some of my favorite cells, which are dendritic cells. Now, look at that picture. Doesn't it? It looks a little bit like a carnation, doesn't it? Now, actually, if you could spin that around and see a three-dimensional version of it, you realize that all these leaflets, they're really, they're more like arm extensions. They're dendrites. They're sticking out. And where would you have those dendrites sticking out? Well, the dendritic cells would be hanging out right under the surface of the respiratory mucosa, extending their arms right under the surface of the gut mucosa, extending their arms and basically sampling everything that goes by, all the food that goes by in the gut, all the microbes that are going by in the gut, in the lungs, the pollens that are going by, the dust particles that are going by, the microbes that are going by all the time. So The dendritic cells are the ultimate samplers, and they are the foremost professional antigen presenting cells. They're also referred to as immune mentors because they are the innate immune cells that really mentor the acquired immune cells and help the acquired immune cells determine their identity, what direction they're going to go down. We have dendritic cells at all bodily surfaces. For example, in the skin, there's cells called Langerhans cells, which are now known to be a type of dendritic cells. It's really up to the dendritic cells to detect the presence of a microbial stranger and at the same time to determine their level of potential danger. So the dendritic cells are all about recognizing dangerous strangers and responding appropriately. And they can respond in two different ways. They can respond with cell-to-cell contact, okay, through an immune synapse, and I'll show you a picture of that. Or they can respond indirectly by producing certain cytokines. And it's not just one cytokine, it's a signature of cytokines. It's a certain mix of cytokines that tells acquired immune cells and other cells what to do next, hence the mentoring concept. Like all innate immune cells, dendritic cells are initially in a dormant state. They have to be primed by exposure to a pathogen and then exposure to a cytokine. They also interact with tissues. They're always sampling gut epithelium. So they know what's normal, and they normally would exist in a tolerant, dormant state. Okay, but don't be fooled by thinking that dormant state is inactive, right? It takes a lot of effort to maintain that dormancy. Now, the type of priming that the dendritic cell receives will result in maturation. So that cell will mature down certain pathways, and that will affect what gets displayed on its surface. The way that I think about it is kind of like being at a bar and having a cocktail waitress come up with a tray. Cocktail waitress got this tray, and I remember the old commercial, cigars, cigarettes, Tipperillo, etc. It's like the cocktail waitress comes up and it says, "This is what I've got on my tray." Well, that tray is the major histocompatibility complex, the MHC. Specifically with immune cells is the MHC type 2 complex. So if the dendritic cells say ingest gluten, then it phagocytoses that gluten. It grinds it up, displays fragments of that on this display platter, and then it shows it to a naive T cell. So it interacts with that naive T cell. We see that on the right, that little red dot would be the gluten. It shows it to the naive T cell, and then if it also sends it inflammatory signals, that will tell that T cell to differentiate down certain pathways and become activated. Over time, we're learning that these activation pathways are very complex. Used to be thought that it was primarily T helper cell type 1 or T helper cell type 2. That evolved to understand that there's also T regulatory cells, which are more suppressive, Th17 cells, which are inflammatory. And now we know that there are even more, and I'll show you a picture of that in a minute. We also know that dendritic cells can activate killer T cells, cytotoxic T cells, and turn them from being dormant cells into active cells. So here's a picture of that immune synapse in the center, that big cell, that's the dendritic cell. And you can see it's a really complex, kind of freaky looking cell. And it's interacting with a T lymphocyte on the left. There is a lot going on at that synapse. A whole lot going on in that synapse. Because that synapse is really where the mentoring goes on. That's where the innate immune cell system communicates with the acquired immune system to say, okay, guys, here's where you go from here. This is the next path in our timeline. Our timeline from this kind of non-specific, broadly induced response to a very specifically induced response with T lymphocytes and B lymphocytes and antibodies. Okay, so we know that once a dendritic cell has become very mature, that it doesn't just have one of these platters on its surface. It has these platters all over its surface. So it can actually bind to several T cells at the same time. And that's what you see in this picture is the dendritic cell in the middle, and it's binding with 3 different lymphocytes. Well, it can actually bind with a lot more. I shudder to say it, but the dendritic cell can be quite promiscuous, right? This is a little bit of an X-rated slide that, you know, it's conjugating with multiple other cells at exactly the same time. So these display patterns, the major histocompatibility complex patterns, can occur multiple times on the surface of the cell. Dendritic cells are very, very versatile cells, some of the most versatile cells in the immune system, which is, you know, interesting considering they weren't discovered that long ago, right? And when they were initially discovered, it was thought that they really played a minor role in immune function. And the researchers that found them really had a hard time convincing the immune community, the immunologists, that this was something they should even be thinking about. And now they really have come front and center, as I said, for their role as a mentor cell. So they're involved in tolerance, antigen capture, differentiation of T cells, cytokine production, peripheral tolerance, etc. They really are central to making, for the immune system to make the decision about which path it's going to go down. Now, let me just throw something out to make you think. When dendritic cells interact with a non-dangerous stranger like a probiotic, such as Lactobacillus acidophilus or Bifidobacteria bifidus, it recognizes the molecular structure as being bacterial, but there's no danger signals. So that's going to cause the dendritic cell to differentiate down a different pathway than it would in a person with celiac disease, whose dendritic cells are going to engulf gluten and become highly inflammatory, resulting in a breakdown of tolerance by activation of T lymphocytes and antibodies against surrounding tissue. So that initial interaction between that antigen in a predisposed cell is going to make all the difference in the world which direction that dendritic cell is going to go. Several times here, I've mentioned pattern recognition. I'll call these pernicious patterns. Maybe that's really not being fair. There's nothing pernicious about the patterns. They're simply patterns that are present in the microbial world. And also, they're present in our own tissue. They're all just molecular patterns. But the innate immune system is programmed to recognize whether a pattern is foreign or not. Okay? And that really gives way to this concept that the innate immune response is almost always triggered by the presence of a molecular structure. And this is an incredibly useful concept because if you see somebody with an inflammatory condition, especially one that's recurrent or persistent, then the $1 million question to ask in functional medicine is, what's the trigger? And at first, that might seem like a question with infinite numbers of answers. But they're not infinite numbers because there's only a set number of molecular patterns that can really activate the immune response. The thing that I want you to remember, and that's what makes this one of the most important slides of the entire presentation, is there are really four major categories of immune triggering structures: microbes, foods, trauma, and toxins. Remember that. Microbes, foods, trauma, and toxins. Come back to that over and over and over again. Here's a patient who's got a persistent rash or inflamed joints or wheezing or a neurologic issue. What's triggering it? Think, could it be a microbe? Could it be a food? Could it be some kind of trauma I'm not aware of? Trauma from say a toxin that they're eating that they don't know about. Or maybe it's electromagnetic trauma that's not immediately obvious. Or perhaps they're getting a heavy metal or arsenic in their food, right? So this can take you off in a lot of different pathways, but it helps narrow down the field so you have some idea of where you're going. For microbes, the pattern we're talking about are called pathogen-associated molecular patterns or microbe-associated patterns, PAMPs or MAMPs. For foods, examples would be gliadin, which we know is inflammatory for everybody. That doesn't mean that everybody has to avoid gluten, but we all have at least a mild inflammatory reaction to it. And that's why when people have a chronic gut problem or a joint problem, et cetera, one of the first things to try is a gluten-free diet. Not because we're always looking for celiac disease, but simply because this is an inflammation-inducing molecular structure. There's advanced glycation in products, glycoalkaloids found in certain foods, pathogen-associated molecular patterns that have been found in processed foods and men's foods, beta-glucans. Again, beta-glucans, not necessarily good or bad, just molecular structures. I'm not going to call them pernicious. I'm simply going to say they're things that our immune system recognizes. I've already mentioned different types of trauma. It can be mechanical, heat, chemical, ultraviolet, or ischemia, simply shutting off blood supply to tissue, leads to cell death. Cell death causes cells to explode. And when those cells explode, they release their mitochondria, their nucleic acids. We shouldn't have nucleic acid circulating in the bloodstream. Circulating nucleic acid are a big inflammatory trigger. And then toxins. I mentioned arsenic as an example. So this is a really central concept to understand. So what we see in this picture here is the green, a cell membrane, and a pathogen associated molecular pattern, a stranger signal that's landing on a pathogen-related receptor. And that generally releases a series of changes inside of the cell, a change in the configuration of the proteins, the transmembrane proteins in the cell. And when that configuration changes, that sets off a cascading process inside of the cell that I will talk about in part 2 of this lecture in some detail, because it leads to activation of a transcription factor called nuclear factor kappa B. So let's get back to pathogen-associated molecular patterns, or PAMPs. These are conserved motifs abundant in microorganisms. So people ask me about a motif. Think about Beethoven, Fifth Symphony. Ba-ba-ba-bum, ba-ba-ba-bum. Now, you will hear that same motif repeated over and over and over again throughout that symphony. You will see molecular motifs repeated over and over and over again in the microbial world. They're typically part of the structure or the DNA of those microbes. And we have in our innate immune cells, and plants too as well for that matter, they have the ability to recognize these motifs. In fungi, we have zymosan, peptidoglycans, glucans, and I'll talk about glucans and beta-glucans in particular in some detail in the immunomodulator lecture. We have mannins, which can stimulate an immune response. In bacteria, endotoxin, teichoic acid, flagellin, myramyl dipeptide, just an example of a few. In viruses, DNA and RNA, both single and double-stranded. Now, these structures are essential to the existence of the pathogen. Okay, they're typically cell wall components, but also can be part of the nucleic acids of the cell, et cetera. So there's something that the microbe cannot live without. And the most important thing to understand is that PAMPs are absent from vertebrates. So when they show up, they provide a clear signal for the presence of strangers, of foreign invaders, just enough to wake up those dormant immune cells so that they know they should be on the alert. Hopefully, if there's no danger signals, there's a tolerance response. But if there's a dangerous stranger, then the immune system is going to go full bore in its response. Here's the danger response that I keep talking about, the DAMPs, damage, danger, associated molecular patterns. Typically, these are sterile molecules. They're non-infectious, and they're produced by damaged cells. Some of the best characterized are fragments of mitochondria. If a cell breaks apart and releases its mitochondria into the bloodstream, that is a major no-no. That is a big signal that damage is going on, and that really activates immune cells. Similar with DNA fragments or RNA from viruses. Okay, RNA from virus can be a danger-associated molecular pattern, even though it's not part of the damaged cell, but it's going to overlap with these same receptors. What about ATP? You don't want ATP circulating around in the bloodstream. You don't want uric acid circulating around in the bloodstream. So all of these are big red flags. Danger associated molecular patterns. So stranger plus danger, that's really the big signal. Damage associated molecular patterns initiate and perpetuate an inflammatory response. When DAMPs bind to receptors, that increases production of pro-inflammatory cytokine. And one thing we'll talk about in part 2 of this lecture is inflammasomes. Inflammasomes are these big protein structures inside of cells that are activated by what's called the NOD2 receptor, intracellular pathogen-related receptor. Now, here's something that I find very interesting, is that the same receptor that's binding to damage-associated molecular patterns also binds to advanced glycation in products, sticky buns, and misfolded proteins, proteinaceous garbage that builds up inside of cells is a part of the normal course of the protein production of cells. Cells are making proteins all the time. A lot of them don't fold properly, and they accumulate inside of the cell. If they get released into the bloodstream or into the CSF, those misfolded proteins, say misfolded amyloid, is highly inflammatory. And it's highly inflammatory because it's binding to the same type of receptor that's binding to DAMPs. It's the identical intracellular pathway that's being activated. So inflammatory foods or damaged cells are activating the same immune response. That is a really profound concept, and I'll show you some data on that in part 2 of this lecture. So here we see in the schema a cell membrane with a pathogen-related receptor that's a transmembrane protein. It's recognizing, say, viral antigen. In the case of the SARS-CoV-2, it might be recognizing a spike protein, or maybe recognizing RNA, and that leads to NF-kappa B activation. Similarly, damaged or stress tissue, it's going to activate pathogen-related receptors. Somehow, and I want to say this, we don't have all this worked out. What's going on inside the cell is incredibly complicated. How does the cell sort out all these signals? I really wish I knew. I really wish immunologists knew. We're starting to get some idea of the pathways, but they're very complex. The best I can say is the cell knows. The cell is able to figure out a stranger signal versus a danger signal and somehow mount a response that's appropriate to the level of threat. And I want to keep saying that over and over again. A healthy immune system mounts a response appropriate to the level of threat, okay, but not inappropriately. That's a healthy immune response, okay? And this is how that works, by interacting with these molecular receptors. This is from an article by Polly Matzinger that shows exactly how complex the immune response is. You see, it distinguishes between self and non-self. That's a starter. And immunology all used to be based on that one concept. All the immune system does is recognize self versus non-self. Polly Matzinger came along and said, no, the immune system recognizes danger, okay? But the immune system also seems to be able to distinguish certain types of infection, right? So every one of these molecular structures or combination of molecular structures is going to elicit a very specific kind of response. And we're just beginning to work out exactly how that works. So what are the receptors that are recognizing these patterns? Well, most of them are glycoproteins or lectins. They're found either on the cell membrane, they're transmembrane proteins, or they're found in endosomes, endoplasmic reticulum. They're found inside of cells as well. Pretty important concept. Why would you want them inside of cells? Because if you got a virus that's reproducing inside of a cell, then you want a receptor that's going to recognize that, okay? As I mentioned, they're germline encoded. When we talk about this ability to recognize PAMPs, this is programmed into the DNA, the germline encoded, and there's at least 1,000 different structures that we have the built-in ability to recognize without any prior exposure. They should not react to self-antigens, okay? There is a built-in mechanism to keep that from happening. And they should be less reactive or non-reactive to commensal. Now, this is somewhat theoretical. A commensal, or let's say Lactobacillus acidophilus, which I mentioned. You don't want your immune system reacting to probiotics. And in most of the cases, they don't. But occasionally, a person does have an intolerant immune system that does allow that to happen. And as I've said repeatedly, to get a full-on activation of the inflammatory cascade, you need additional danger signals like mitochondrial fragments, uric acid, ATP, anything that's supposed to be inside the cell if it's outside of the cell, or even if it's supposed to be in a compartment inside the cell and it's released into the cytosol, that can interact with a pathogen-related receptor and cause a problem. So here we see different types of PRR, pathogen-related receptors. On the left, this is the RAGE receptor, the receptor to advanced glycation end products. I call this the sticky bun receptor. So you can see that tiny little image of sticky buns, croissants, cinnamon buns, cinnamon rolls, all these foods that taste so good, that smell so good, are so highly inflammatory. The second receptor we see is a scavenger receptor that's responding to lipopolysaccharide. Then we see a toll-like receptor, TLR, which I'll talk about in a little bit more length, responding to a bacterial pathogen. Then we see a receptor, the CD36, that's responding to a hamburger. And again, all of these things are eliciting an inflammatory response. So let's talk about one specific type of PRR, toll-like receptors, TLR. Interestingly enough, they're very similar versions of this in Drosophila melanogaster, the common fruit fly. That's where they were first discovered. And when they were discovered, the researcher used this phrase, "Das ist ja toll." It's an amazing thing that we found. Okay, so that's all toll means. Now we know there are a number of them. They play a critical role in the innate response against bacteria, viruses, and fungi. And where do you find them? On and in immune cells, dendritic cells, macrophages, mast cells, and T lymphocytes. All of them lead to activation, change in configuration of intracellular proteins, which sets off a kinase cascade that turns on NF-kappa B and triggers the immune response. The first of the toll-like receptors to be identified was TLR4. In fact, the researchers that identified it got the Nobel Prize for that. It was that important. TLR4 recognizes endotoxin, lipopolysaccharide, among other things. Then we began to realize there were a lot of other toll-like receptors. I'm guessing, can't say this for a fact, but when they discovered TLR4, they probably didn't call it 4 because they didn't know it was number 4. It was simply the first of the toll-like receptors. And I've seen this evolve over time. Initially, hey, big discovery, toll-like receptors. Now we're finding lots of them. TLR2, TLR9, TLR3, and TLR7 specifically seem to be the ones that recognize intracellular SARS-CoV-2, right? So if that virus is replicating inside of cells, it's interacting with those toll-like receptors. And if you have a huge viral burden of, the nucleic acids, then you may have an overactivation of TLR3 and TLR7 that leads to hyperinflammation. This is one of my favorite slides. Basically a zoom in kind of slide. On the top left-hand corner, you see epithelial cells of the gut. You see the villi, and on the left side of those villi are bacteria. Okay, and what do you have under the surface of those enterocytes? An antigen-presenting cell. That antigen-presenting cell has sent up its dendrite that samples the bacteria. Now what we see in the bottom right is the cell membrane of that antigen-presenting cell. What's going on there? Well, there's a toll-like receptor that has recognized the antigen, the molecular structure on the surface of that bacteria. Let's just say that it's endotoxin, lipopolysaccharide, and there's this Rube Goldberg type reaction inside of the cell where you activate that transmembrane receptor that activates an intracellular protein called MyD88. That sets into motion a series of kinase reactions, which ultimately will phosphorylate inhibitory kappa B. And when it's phosphorylated, that allows for liberation of NF-kappa B, which then gets activated, goes to the DNA, turns on the DNA to make TNF-alpha. So I've run through that really quickly. We're going to come back to that mechanism over and over again because it's really central to understanding the inflammatory response. Here's a very practical implication of all this. Is that our gut microbiota, which I talk about in some detail in the pre-course webinar on the gut microbiome, is a primary source of lipopolysaccharide, right? This is normal. We've already got E. coli and other gram-negative bacteria which have endotoxin on their cell surface, okay? When we eat a super energy-rich meal that is high in fat and refined carbohydrates, the super fatty meat combined with the French fries, and that's pretty important to understand that. It's not just the fat, it's the carbs in there, the potatoes. When you put all those together, you produce these chylomicrons, And the chylomicrons will ferry lipopolysaccharide into the bloodstream. And when that lipopolysaccharide gets into the bloodstream, it binds to toll-like receptor 4 that's on the surface of immune cells, fatty cells, endothelial cells, and liver cells. And this leads to what's called metabolic endotoxemia, right? We all know about toxemia that's life-threatening with a collapse in blood pressure, septicemia, etc. What about a very, very low-level kind of toxemia? Metabolic endotoxemia, simply from eating inflammatory foods. We know that one of the consequences of this metabolic endotoxemia is insulin resistance. Well, this is a way that this chronic low-level inflammation, this inflammatory state, can lead to obesity, type 2 diabetes, fatty liver, and heart disease. In other words, this is an entirely different mechanism than simply saying person eats sugar, their blood sugar goes up, that leads to insulin resistance, and that's what causes all these problems. This is how an inflammatory diet can go down a parallel pathway and create the same kind of phenomenon. So again, let's get back to this overall schema. Now you're starting to see why it's important that we understand that the innate immune response is really responsible for the overall immune response. If a person's eating inflammatory foods, then that's going to program the innate immune system towards an inflammatory pathway. Okay, so that's the interface, the initial interface in our timeline, the initial interface with the outside world, what we eat, and the microbes in our gut are going to influence the innate immune system. This it's a non-specific, but that's a little bit of a misleading concept. It's not really that non-specific. It is specific to certain molecular structures, to certain stranger plus danger structures. So again, we see the innate immune system on the left, and then it all converges on this antigen presenting cell interacting in the center with the activated T cell, which then is going to become differentiated down certain pathways towards differentiated helper cell. The helper cell then influences the B cell to become a plasma cell, or a cytotoxic cell to become a fully activated cytotoxic degranulating cell. Same slide that I showed you before, but hopefully you have a different understanding of that now as we move from innate immune cells through the overlapping cells to the fully adaptive immune cells. Here again is a close-up of that synapse. Lots of things going on at that particular synapse. This is an image which I love from the New England Journal of Medicine. It shows that antigen presenting cell, dendritic cell, and see all those little dendrites going out. And it's showing that there's two major ways that dendritic cell can interact with a pathogen-associated molecular pattern. It can interact at the top through a toll-like receptor, which turns on the inflammatory cascade, or it can interact through an endocytic pattern recognition receptor, which leads to phagocytosis. If it eats that pathogen, or as I mentioned, something like gluten, an inflammatory food, it eats that food, it grinds it up, it puts it on the platter, the MHC type 2 class 2 molecule. It puts it on the platter and it presents that peptide to a naive or quiescent, a quiet, dormant T cell. Now, I want you to look right there in the center. You see the MHC class 2 molecule, the peptide in the center that's being presented, the T cell receptor, the TCR on the right. That's just the beginning of this process. In addition, you've got to have all kinds of other signals, co-stimulatory signals that the dendritic cell produces. In this case, it's a protein called B7 that binds to a receptor called the CD28. These send additional signals to the T cell that tell it whether to respond or not respond, whether to become inflammatory or immunosuppressive. So it's all in these other signals that come along with the display. It's not just the display, it's the secondary signal, signal number 2. This is a little bit more elaborate picture of the same thing. Again, the dendritic cell on the left, naive T cell right in the very center. And you can see all of these different locks and keys that are occurring at this innate synapse. So, immune system basics. How does a virus lead to adaptive immunity? Well, the virus infects and replicates in the epithelium. It's engulfed by a dendritic cell. The dendritic cell then travels to a regional lymph node. And in the regional lymph node, the dendritic cell grinds up that virus, displays it on that platter that I mentioned, interacts with the naive T cell. That naive T cell then becomes activated, becomes a T helper cell. And that T helper cell then starts making cytokines that will influence other cells, will influence cytotoxic T cells, will influence B cells until B cells become increasingly activated and differentiated. Okay? Interestingly enough, this is the same kind of thing that happens with vaccines. If we have an mRNA vaccine, that mRNA is going to be taken up by phagocytic cells. We don't know exactly which phagocytic cells, but I think the assumption right now is that it's macrophages in the tissue. So a person gets an mRNA vaccine. That mRNA is going to be chaperoned into the cell by lipid nanoparticles. So they basically form a shield that allows it to cross the membrane, gets inside of a macrophage. The macrophage then starts making the spike proteins as directed by the mRNA vaccine. The spike proteins then get displayed out on the platter, and those spike proteins then are sent as signals to T cells to be activated, so that now you have specific T helper cells that will recognize spike proteins. Or there's going to be activation of B cell to make specific antibodies against the spike protein. So now hopefully you're getting a better understanding of exactly how the adaptive immune system works. The adaptive immune system is only found in vertebrates. Okay? And the whole central concept of adaptive immunity is memory, is the memory after exposure to an antigen. Well, what kind of memory we're talking about? You know, we have in our bloodstream really millions of antibodies, so all kinds of things that we're exposed to, we keep memory of these cells, but we don't keep zillions of these antibodies around. We keep just enough to provide an initial barrier after re-exposure, and then those B lymphocytes need to proliferate again. So 2 major types of acquired immune cells, T lymphocytes, which mature in the thymus, and B lymphocytes from the bursa fabricis, which is where the B comes from. They're made in the bone marrow, but that's not the B is named after. So both T cells and B cells have a very interesting origin, which is that during the maturation process, they randomly generate receptors simply by rearranging the DNA, which rearranges the proteins on the receptors. And when you rearrange the proteins on the receptors, that allows for almost an infinite number of abilities to respond to different molecular structures. Okay, so T cells and B cells both start out that way. We make B cell receptors to a huge number of molecular structures. So B cells have this huge numbers of immunoglobulins, but only a few of them get activated. The rest of them stay dormant in circulation until they get exposed to that particular antigen. Again, that's the function of a vaccine. let's come back to this notion of co-stimulatory molecules. We have the first signal. That's the display of the antigen by the antigen presenting cell, a peptide bound to MHC2. Okay, the T cell receptor recognizes the antigen, but it's not necessary that anything happens at this point. Okay, that's just the beginning of the conjugation process. But then we have a co-stimulatory signal. Now, if the T cell is going to be activated, then a typical co-stimulatory protein would be the B7, CD80 or CD86, collectively called B7, that engage a receptor on the T cell. So a number of years ago, it was thought, hey, this is a great way to activate the immune cells. So let's come up with an antibody, a super agonist, monoclonal antibody, an MAB, to CD28. That way we can activate T lymphocytes at will. We can give people a shot, we'll activate their T lymphocytes, and maybe we'll have a cure for cancer. Guess what? When they injected people with these, even though in animal studies they hadn't seen any problems, no side effects, etc., when they injected humans with these, their cytokine responses went through the roof. And this is really one of the first descriptions of what was called a cytokine storm. Okay, and the cytokines started killing organs, and these people got really, really sick. Got really, really sick, totally unexpected. I don't think that anybody died from this, but I think people came close to death from this. So that helped us realize, this is an area we've got to be really careful if we're messing with this immune synapse between the dendritic cells, the antigen presenting cells, and the lymphocytes. We start messing with this, all kinds of unexpected things can happen. Now we think of these receptors, these interactions, as immune checkpoints. Okay? We've actually got one set of receptor interactions that are an on switch, and then we have other ones that act as an off switch that modulate tolerance. Okay? And this dampens the inflammatory response. So when I began this lecture, I told you that whenever you turn on the inflammatory response, we also start turning it off. The same thing is happening in this interaction between the innate immune system and the acquired immune system. You turn it on, and then immediately with this immune synapse, you start turning it off. This is an immune checkpoint. Okay? And this requires, the binding of the T lymphocyte receptor to that peptide that's being presented, but also a co-stimulating ligand. So that's where it begins. And then the third step is this off switch. So you turn it on, but immediately you're turning it off. So it's kind of a give or take, a push or pull. Here's a picture of this. I know this can be a little bit confusing. The bottom line is that this has led to a whole class of drugs that's used to treat cancer. So let's start in the middle. Activated antigen presenting cell. You see that, again, that major histocompatibility complex, the MHC, presenting a peptide to the T cell receptor. You've got the B cell protein, that's the co-stimulatory molecule turning on the T cell. If that's all that happens, then the T cells will proliferate, differentiate. Now, let's go to the top. If the antigen presenting cell, say, displays that peptide, but doesn't display the co-stimulatory molecule, the naive T cell doesn't respond. Okay? But let's go down to the bottom. You've got the initial signal, signal 1, the peptide, signal 2, the co-stimulatory molecule, and then what you've got is an inhibitory molecule. And that inhibitory molecule, the CTLA4, if that gets turned on instead of the CD28, then that keeps the immune cell from going full bore. Okay? So that would be a good thing if you want your immune cells to be fully activated and fight off cancer. And that's why this has been used in melanoma. for example. Here's the problem. If you activate that CTLA4, which is the down regulator, if you turn on the down regulator that is supposed to be, if you're inhibiting the down regulator, then you can actually get something that looks like autoimmune disease, actually is autoimmune disease. So you can get people who develop type 2 diabetes, or in the case of one of my patients who got a CTLA4 inhibitor, he got full-blown serious colitis. So we're just beginning to learn what happens when you use these checkpoint inhibitors. When you turn off, if you're inhibiting the switch that turns off the inflammatory response. Let's get back to that whole immune synapse again. Here we see a dendritic cell. The dendritic cell is interacting with a naive T cell. And the initial model was that that naive T cell could become what's called T helper 1, involved in tumor immunity, intracellular pathogens, or Th2, involved in extracellular pathogens. That model got expanded to include T regulatory cells, which are immunosuppressant, and highly inflammatory Th17 cells. Okay, so this model, I can tell you, is getting expanded day by day. You know, what's still true is at the top, we've got this interaction between dendritic cells and naive T cells, but there's a lot of different pathways that these naive T cells can go down, and we're just beginning to understand what all those possibilities are. In fact, here's an example of some of the newer subclasses of T helper cells that are starting to appear. Th9, Th22, TFH that produces interleukin 21. My guess is that when I give this talk in another year or two, we're going to continue to add T helper cell subsets. So the important take-home message is really what's going on at the top, that the naive T cells can go down a number of different pathways, can be more or less inflammatory, really depending on the initial exposure to the pathogen. So again, this is based on the older model, but I think we're still with the same concept that we have different types of T helper cells, and the balance between them can really determine whether we have tolerance, if everything is balanced, or if we have allergic inflammation from, say, too much Th2 activation, or whether we have chronic inflammation from too much TNF alpha. So it can go down a lot of different pathways. Th17 cells, a particular type of T helper cells, are one that has really come into interest in the last few years. Again, it was initially thought, well, we either got the choice between 1 or 2, and then 17 kind of emerged. Th17 makes a number of different highly inflammatory cytokines, IL-17, 21, and 22. Okay. We now know that Th17 cells are involved in a lot of autoimmune diseases. For example, psoriasis and psoriatic arthritis is one that's really been focused on a lot. Okay. What induces going down that pathway? Interleukin 1 beta. Interestingly enough, that is increased by inflammasome activation. And in part 2, I'll talk about what turns on inflammasomes. Okay. But it's activation of toll-like receptors, really certain ones, especially fungi, that turn on Th17. So Th17 cells are thought to make interleukin 17, and those have been implicated in autoimmune disease, cancer, allergy, Lyme arthritis, cardiovascular disease. Fortunately, there's a drug, secukinumab, a monoclonal antibody against interleukin 17. Well, I say fortunately and unfortunately. For the most severe cases, this may be appropriate. Okay, but it's a very expensive drug. And as you can imagine, there's side effects. So we're looking for natural alternatives. Interestingly enough, you do see downregulation of Th17 cells by vitamin D, by vitamin A, by zinc, by statins, by flavonoids like quercetin, licorice root, and an herb called thunderduke vine, which has a compound called triptolide. So there's a lot of interesting research that's emerging around this particular acquired immune cell subset, the Th17 cells. Last but not least, T-regulatory lymphocytes, okay? I like to say these are the good guys. Well, they're good guys in a lot of situations, okay? They're immunosuppressive cells. They're about 10% of the T cells that are typically in circulation. They're induced by transforming growth factor beta made by dendritic cells. So dendritic cells are mentoring those naive T cells to become regulatory. And we think that's what happens when a person is taking probiotics or when they have a predominance of healthy bacteria in their gut that's activating dendritic cells to make more T regulatory lymphocytes. And that seems to be mediated through the toll-like receptor 2. The functioning role of Treg cells is to modulate all these different lymphocyte subsets, Th1 and Th2. I should also add Th17. So the Tregs kind of provide the super cop role there. So the net effect of Treg cells is to maintain tolerance by dampening immune response to ingested antigens. And we know that's the case because if you genetically program mice to not make Treg cells, they get spontaneous autoimmune diseases. So that's generally a good thing. We want to have more Treg cells. We want to have healthy amounts of Treg cells. But bear in mind that cancer cells can hijack Treg cells to function as immunosuppressants. So it's not always a good thing. I've not seen any literature, however, that says that probiotics can upregulate Treg cells and that could lead in a way that would lead to cancer. So not to worry about that. Generally, it's a hijacking phenomenon where the cancer hijacks the genetics of the lymphocyte to make it more functional as a T regulatory cell. So here we see that naive T cell, the Th0 at the top, that it can either become a Treg cell, Th1, Th2, or Th17, or any of the other inflammatory subclasses. And what we see is that, again, the Treg cells kind of sit in the center and they control autoreactive T lymphocytes. So that's generally a good thing. We need them. We want to induce them. So we want to find out what foods we can eat, what probiotics we can take that will keep our Tregs healthy. And that leads to this concept, which I think this is a beautiful article. Regular Tai Chi Chuan enhances functional mobility, and CD4, CD25, those are the cell markers on reg cells, enhances the activity of T regulatory cells. So it's not all about diet, necessarily supplements, simply healthy lifestyle practices will lead to more activation of these generally beneficial T regulatory cells. So this final slide is a lead-in to where I'm going to go with part 2, which will actually be the formal beginning of the module. So I want you to think about inflammation, learn about inflammation, and get ready to take a deep dive into the molecular processes involved in inflammation. With that, I'm going to bring this particular lecture to a close. Thank you very much.